As you can see, I'm standing here in the wire room, but it's empty. That will be very different on the 9th of March, as on that day we will be celebrating our Dies Natalis, as we do every year. This year the Dies Natalis will especially focus on transitions which are so necessary in the field of energy and climate change, in the field of food, in the field of agriculture. And science plays an important role. Not just applied science, but we will be focusing especially on the role of fundamental, basic science in achieving those major transitions. So I would like to welcome you very much on the 9th of March, here exactly in this room.
I'm Jeremy Harbinson and I work in the biophysics group of Wageningen University. My specialism is photosynthesis. In fact, I really love photosynthesis. Photosynthesis is a process we should all know about. It's the engine of crop productivity. It uses light to convert carbon dioxide into more plant. Don Ort, a great scientist from the United States, is going to give a speech at the Dies Natalis of Wageningen University, where he's going to explain how our basic knowledge of this process has been used to improve the productivity of crop plants, and in doing so, offering a means for guaranteeing future food security.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in the forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
As you can see, I'm standing here in the wire room, but it's empty. That will be very different on the 9th of March, as on that day we will be celebrating our Dies Natalis, as we do every year. This year the Dies Natalis will especially focus on transitions which are so necessary in the field of energy and climate change, in the field of food, in the field of agriculture. And science plays an important role. Not just applied science, but we will be focusing especially on the role of fundamental, basic science in achieving those major transitions. So I would like to welcome you very much on the 9th of March, here exactly in this room.
I'm Jeremy Harbinson and I work in the biophysics group of Wageningen University. My specialism is photosynthesis. In fact, I really love photosynthesis. Photosynthesis is a process we should all know about. It's the engine of crop productivity. It uses light to convert carbon dioxide into more plant. Don Ort, a great scientist from the United States, is going to give a speech at the Dies Natalis of Wageningen University, where he's going to explain how our basic knowledge of this process has been used to improve the productivity of crop plants. And in doing so, offering a means for guaranteeing future food security.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in the forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
As you can see, I'm standing here in the wire room, but it's empty.
So if you're in the neighborhood, you're allowed to do that? Yes. So my colleague was
Het ging toen al dat ik op heel
Boys.
Ja, precies. Voor jou speciaal voor jou. Ja.
Je zal het door dat we niet toevallig nastropen of zo.
Jacques, hallo. Dat is allemaal goed gelukt, hè? Jacques, hallo. Kijk, het is het comité hier zo. Ja. En jullie gooien de kamer daar. Niet buiten. Een mooie stranddag. Ja, het is weer anders dan net, hè?
Hallo. Nou, gaan hè? Nee, we hebben heel veel dingen op de lijst staan, maar die wel een toog, die hadden al
Ja, ik ben niet te perfect zoals. Ik kan alleen niet verder pennen naar links. Ja, ik kan dus niet verder naar links.
Please be seated. Distinguished colleagues, representative of different embassies, Mayor, Mr. Mayor, Mr. Colleague from the alliance which we will be establishing shortly this week together with the University of Eindhoven. You are from Utrecht. Ladies and gentlemen, parties of different kinds, students, welcome all to this 102nd Dies Natalis of our university and research. We are very welcome to have you all here at this little bit strange time due to, of course, the coronavirus. Of course, you can imagine that we also discussed a little bit internally whether we should continue this, but I think it would be strange not to do so, because that would also mean that we have to postpone all our education, and that would be a little bit too drastic. And also we consider it still pretty safe, and your presence here also showed that that was indeed right so. Transitions, if I'm correct, yes, are a fashionable term these days. We see them emerging everywhere. Certainly the last couple of years, but maybe a little bit longer, 2 decades ago, the concept of transitions is very much prominent. We see transitions in relation to energy and climate change. We see transitions related to mobility, to food and agriculture, to industry, etc. Transitions then stands for the need to have systemic change. Systemic change is a little bit the opposite of incremental change. Because these systems do no longer fulfill the requirements which we would like them to fulfill, the need for transitions is there. And usually these requirements are very much linked to sustainability at the core of our university. Science and scientific institutions have very well understood the need for knowledge, for further action on these transitions. And many of the universities have indeed developed all kinds of programs. And this, some of you will know this picture is from our strategic plan, which was launched a little bit over a year ago, where indeed the need for transitions, the need for knowledge for transitions was very prominent there. There's even now such a thing as transition science, which estimates and underlines the role of transitions in society and in science. What I would like to do in this introductory talk is to go over two aspects of transitions in relation to science. The first one I will call science in transition, and the second I will call science for transition. So I'll start with science in transition. In 2012, in San Francisco, the DORA Declaration was signed, the Declaration on Research Assessment. That declaration, signed by many universities, research funding agencies, scientists individually, from all over the world, basically said that we should have another look at how we assess excellent science, how we assess excellent scientists. And one of the key objects of that declaration was to play down a little bit the role of journal impact factors and some of the other metrics which were there in assessing excellent science. Last year, the Association of Dutch Universities, the VSNU, also published a report which was called Room for Everyone's Talent. And that was in the same line of what is now called Science in Transition, a movement in science among scientists. And in that report, a number of guidelines, a number of ideas were put forward, and all related to how we should assess scientists and academics. There are 6 points I want to go over a little bit. Not so much to say that is how we should do it, but basically to put here before the audience, as we will be going the coming year through an organization-wide process in discussing what is now exactly excellent science and what are excellent scientists and how should we assess them. The first point, which was also put forward in the report by the VSNU, the Dutch Association of Universities, is that the aim we always want to achieve in assessing scientists is excellence. We want to put forward and to further excellence in science. There can be no doubt on that. The legitimate question is, of course, constantly, what is now exactly excellent science? Excellence is, of course, a very diverse term. You can be excellent in very many ways. That has meant that some of the scientists has said, well, I can also set my own goals to be assessed along, and that excellence can be a variety of goals. Or to put it in Feyerabend's works, anything goes. I think that goes a little bit too far. The same is the discussion on tenure track, which is now also very vivid in our organization, where some also say, well, we are about to end the tenure track system. Well, let me then also paraphrase Mark Twain's work, and that is that the report on the end of the tenure track system is greatly exaggerated, or the death even of the, as Mark Twain said. So I think that's the first point. We will continue to go for excellent science. We will also use, to some extent still, metrics for that, but only where they are very useful for that, and in addition to many other ways of trying to assess ones. The second point is that very often excellence in academics is only focusing on education, on research. And I think the point was very well taken in the report of the VSNU that education is as important in our kind of institutions as research. And I think in Wageningen we have understood that message, at least to quite some extent. We are also looking at education in our research and development talks every year. We are looking at education in the tenure track system. We have just established a separate path and a career path, an education career path for that. And of course, the best token is, of course, that we are doing very well in education, being already 15 years in a row the best education university in the Netherlands. Does that already put education on a par with research? Well, probably not. But I think in revising the system, we don't start from scratch here. The third point is, of course, that even beyond research and education, there are other dimensions of excellence. Value creation is an important objective which we have here in our organization. Leadership is an important one. Open and citizen society is an important one. And you see the amount of dimensions emerging and increasing. Can we expect all our scientists to be excellent in all those dimensions? No, I think that would be a little bit too much. We are already touching upon a quite high level of workload, as we see in all kinds of surveys, so we should not increase that too much. Would then be to go for only one of the dimensions, so have only excellent researchers, excellent teachers, excellent managers, excellent open scientists, etc. I think also that would not be a wise avenue to take. The strong point of Wageningen has always been in combining research and education with value creation and certain kind of management tasks. So it will be a balancing act which we have to do in combining these different kind of dimensions. And one of the solutions could, of course, be is no longer look at individuals as such, but also look at teams. And that's, of course, the fourth dimension. Teams are getting increasingly important because science always stands on the shoulder of your predecessors, but also on the shoulders of your teammates. It's increasingly team science and not so much individual science where the frontiers are pushed forward. So we also have to take teams into account in assessing scientists. To some extent, we do that in some cases. In the zwaartekrachtprogramma, the gravitation program of NWO, we look at teams. In the assessment of the graduate schools, we also look at teams which are there. But in many instances, we still look at individuals. In personal grants, we look at individuals. In R&D talks, we look at individuals. In tenure track, we still look at individuals. So I think there will be a major challenge to combine the two types of assessments together, to look both as individual excellence as well as team excellence. Fifth, I think in any system of assessment, we cannot only look inwardly. We have to take the international connotation into account. If we build our system of assessing excellence, excellent scientists, excellent science, we also have to pay attention to what is happening in other universities across the world. We are really an international organization. If we don't pay attention to the outside world, we also shoot ourselves in our foot. For instance, in terms of recruitment, in terms of funding, in terms of our ranking positions, etc., etc. So we need coordination in this new kind of assessment system. We do that in the Netherlands already by the Dutch Association of Universities, but we also need to do that internationally, in Europe and beyond that. So that will be a major effort also. And finally, on still science and transition, assessing scientists cannot be an executive board decision. It's not a one-time decision by the leading management. It will be and has to be a cultural norm-building process. And I mean by that, that we have to do it together. Set together the norms, new cultural norms in what is now excellent science. And we need to do that because we always do that in Wageningen. We never do that from the top, but we always involve process. But we also need to do that because scientists are always the one who assess colleague scientists. In tenure track system, in assessment committees, in review procedures, in judging proposals, etc. So in that sense, we also need to do that together. And because we need to do that together, I've decided, or the executive board has decided to set up a committee headed by our Dean of Education, Arnold Brecht, to develop for the coming years in a wide, organization-wide process, a new system of how we are going to assess and reward our scientists. So I would invite you all very much to join that process and to put forward your ideas on how we can best or better do that. That's for the science in transition. Let me now move to the second part, the science for transitions. As I just said, science has very well understood the need to have knowledge to push forward the transitions which are so needed to move to a sustainable future in the Netherlands, in Europe, and widely. Here you see a graph in how much relatively our organization has contributed to publications in a number of the Sustainable Development Goals, which are the hallmark for transitions, so to say. And you see that's quite a lot. We see that also in our own strategic plan, what I just said. We see also there a strong commitment to contribute as an organization to these Sustainable Development Goals by indeed a certain amount of science. Focus on Sustainable Development Goals or transition processes, on missions, whatever you would like to call them, is of course highly legitimizing science, and that's good for us as an institution, which is also sometimes in debate. But it can also lead to a particular type of science. And here I show you what is called the Pasteur's quadrant. And the Pasteur's quadrant nicely distinguishes 4 different types of scientific research along 2 axes. One axis is the consideration of use, the horizontal axis, and the vertical axis is whether fundamental understanding is at stake. And you see then 4 different types of research. Now basically what you see is that if you have a heavy emphasis on Sustainable Development Goals, on missions, on transitions, etc., that might lead to certain types of these quadrants, especially the right upper one and the right lower one, the use-inspired research and the pure applied research. And it might not so much favor the pure basic type of research. Basically, that was also the finding of a recent report of the Royal Academy of Arts and Sciences in the Netherlands, where they just found out that indeed there was a rapid increase of what they call thematic research, what I would call here in this quadrant applied research, and a little bit use-inspired research. That increased a lot, but it came with a stagnation of pure basic research. If we, however, want to push forward transitions, we need also pure basic research. We need really new fundamental knowledge which are not just incremental increases of our knowledge, of our innovations, but fundamental new types of knowledge, fundamental new types of innovations. And it's especially pure basic research which might deliver us that at least earlier than the other types of research. There are also some good examples in the past how that works. For instance, the internet or the accuracy of GPS systems did not came from applied research, although they are now very widely applicable, but came from pure fundamental research. If you look a little bit closer to our domain, the major breakthroughs in gene editing, like CRISPR-Cas technology, didn't came from research on gene editing, but came especially on research on the immune system of bacteria. Not so much focused on gene editing, but just wanting to know how that immune system works. So basically, the message which I want to convey here is that we need pure basic research. We need not to forget that, not in this university, but also on a national and European scale. That's not always a message which is easily taken by politicians, by university managers, I say for myself, by funding agencies, etc. Because, of course, this goes with large uncertainties. It goes with users which are unknown and thus unheard. That doesn't make it very easy. And usually also the payback time or the impact are rather in terms of decades than in terms of years. So we need really to push to have that on the agenda. There's one but. That should, of course, not go together with a decrease in applied research or in use-inspired research. It's, of course, the combination of these different types of research which gives us the right material, the right knowledge to build those kind of knowledge systems for transitions. We need ecosystems where all 4 kinds of research are present. And basically, you understood already the message. That's exactly what we are trying to do here on Wageningen campus. We try to integrate basic research, with user-applied research, and with pure applied research. And also the unlabeled category, as Stokes said in his quadrant. Well, the best way, of course, to show that indeed this is possible, and this is also happening, is to give a number of examples for that. And of course, one of the examples, which is, I forgot that one, is very close to our heart, is of course on photosynthesis. So I could elaborate a lot on what kind of new, interesting research is taking on these days. Where are the frontiers of photosynthesis research? What are the unknown territories? Where do we need more fundamental science and insight in order to have new applications? Well, that would not be a good idea, because I'm not an expert. So I'm very proud that we have an expert today in the room. It's a great honor for me to introduce to you Professor Donald Oort from Illinois, Urbana-Champaign. And Professor Donald Oort is the Robert Emerson Professor in Plant Biology and Crop Sciences, and already for at least more than 30 years working on photosynthesis research. So I would like to invite you to stage, Donald, and to show us where are the fascinating points of this fundamental and applied research on photosynthesis. Floor is yours.
Well, thank you very much. It's really a pleasure to be here. I was honored to be invited. I suspect that maybe a contributing factor to me being invited is that this year's theme of transitions from basic research into translating into research that addresses some of the world's problems is in some ways traced by my own scientific journey. And so when I got into photosynthesis research, we didn't actually completely understand the mechanisms and all the players. And so in much of my career has been involved in doing basic research, asking fundamental questions using a very reductionist approach. But I think because, for me, I really want to understand what plants do and how they do it. For me, the goal of the reductionist approach was to learn enough to try to start putting it back together and understand things at higher levels of organization, so it could be transitions or translated into addressing some of the fundamental problems that our society faces. And so what I'd like to do today is to talk about work that we've been doing over the last 10 years, focusing on the redesign of photosynthesis to improve photosynthetic efficiency as a strategy to improve crop productivity in the field. And so I want to do three things. One is I'd like to take a moment and step back and make sure we're all on the same page of understanding what the challenges are that our agricultural enterprise globally faces over the next few decades. Then I'd like to talk a bit about why redesigning photosynthesis for improved efficiency is a promising opportunity to try to address this. And then finally, I'd like to give a proof of concept set of experiments that we've done over the past several years that shows that increasing photosynthetic efficiency can lead to increases in productivity of crops in the field. And so the place I'd like to start is here. And so this is the terrestrial global carbon cycle. And what this shows is that photosynthesis removes amount of carbon from the atmosphere every year that's equivalent to 15% of all the CO2 that's in the atmosphere. And so this is a massive biological flux. About half of that is returned back to the atmosphere by autotrophic respiration of the terrestrial biome that does that photosynthesis. And so this is the respiration that's required to grow this terrestrial biome and to maintain it. The other half, which is often called heterotrophic respiration, on an annual basis represents about half of gross photosynthesis and is responsible for supporting all other life on the planet globe. And you can see that this is about 60 gigatons. And so the difference between gross photosynthesis and net photosynthesis is often called, mainly by ecologists, net primary productivity. And I think it's really instructive to look at the biomes that are responsible for this net primary productivity. And so it probably doesn't surprise anyone in this room that the tropical rainforests represent about a third of the terrestrial biome's net primary productivity. Another third is represented by the vast savannas that are deployed around the world. But what may surprise you is that already crop cultivation represents a carbon footprint of about 15% of all the carbon that's taken out of the atmosphere by net primary productivity. And so that means that our carbon footprint to feed ourselves and the livestock that we grow already accounts for 15% of the total carbon uptake. And that's equivalent to approximately what all the temperate forests do. And so the UN FAO, the UN Food and Agriculture Organization, projects that in order to meet agricultural demand, this is demand that's driven both by population increase and by the increasing affluence of some of the most populated countries in the world, we'll have to double in order to meet that agricultural demand. This seems like an enormous challenge, and it is, but I think it's interesting to think, you know, it's already been done once. And so in the period between 1960 and 2005, when population grew about 110%, went from 3 billion to 6.5 billion, global crop productivity grew an incredible 165%, went from 1.8 to 4 billion tons. It's important to understand that 135% of that 165% increase was due to intensification. That means more produce on the same amount of land. And only 25% was due to recruiting more land under cultivation, and most of that was in Africa. What this graph shows is that the land under cultivation peaked somewhere before 2000 and has been declining slowly ever since. And so if we really have to double productivity, I think what this has to mean is we're going to have to do it on the 1,200 million hectares that are currently under cultivation, and probably even less. And so we have to double productivity on a land area basis if we're going to meet this challenge. And so what that means, if we go back to this, is that now we go from a carbon footprint of 15% to a carbon footprint of 25%. The fact that we're doing this on the same land area basis suggests that global net primary productivity has to go up by about 12%. And so this truly is a grand challenge. Unfortunately, crop production is not going in this direction. And so this is data that we took again from the UN FAO, and the way to read this graph is that in the decade ending in 1977, these three crops had a decadal increase of about 30%, or about 3% per year. And so what you can see is that was sustained through the 1980s. By the 1990s, these two small grains started going down, and by 2007, which is the last year that we have complete UN FAO data, you can see that wheat was down to a 0 increase, and rice was down to about a 15% increase. You know, so somewhere between 0 and 1.5% per year. And you project this forward to 2050, 2060, or 2070, and you fall very well short of anything close to the doubling. It's interesting that, for rice, China is the world's biggest producer and consumer. It produces and consumes about 30% of the wheat in the world. It's investing now several hundred times more than it did in the 1970s and the 1980s. A lot's come out of that. And so 1980, they've increased rice productivity in China by 50%. But still, even with those 50% reductions, you see it dropping. And so what that suggests is that those things that were driving yield increases and selected by breeders, the low-hanging fruit is gone, and we really need to be looking at different things. And so what is the cause of this yield stagnation? And so I think it's useful to look at what goes into determining crop yield potential. And so this is a fairly famous equation. It's sometimes called the Monteith equation, and it defines what goes into yield potential. And let me define yield potential first. And so yield potential is the amount of yield that you'll get off of a particular crop that's grown in an area where it's well adapted, it's grown with enough water, it's grown with enough nutrients, pests and pathogens are controlled, as are weeds, and there aren't any other dominating kinds of stresses like heat waves. And so it's really the genetic potential of the crop. And so this genetic potential is constrained by the total amount of solar energy that's available to that crop during the growing season. And so that's all the energy there is to drive photosynthesis, and photosynthesis is what's driving the net primary productivity of that crop. And then that sunlight is acted on by 3 genetically determined things in the crop. So one is the efficiency with which the crop intercepts that radiation, the efficiency with which photosynthesis converts that radiation into plant biomass, and then finally, the decision that the plant makes of how much of that biomass to partition into the part of the crop that we care about, so the wheat grain or the rice grain or the corn kernel. And so when I talked about what happened during the Green Revolution and we had this 165% increase in productivity, it was really driven by 2 things in this equation. One is the interception efficiency. And so breeders got very, very good at selecting for plants that covered the ground very quickly. If the light hits the ground, it's not going to do photosynthesis, it's not going to drive yield. And so now our best performing crops are intercepting about 75% of the total radiation that's available during the growing season. We think the theoretical limit is somewhere between 80 and 85%. And so room for a little bit of increase, but we're certainly not going to drive a doubling based on improving interception efficiency. The other thing you probably all heard about the Green Revolution is this improvement in harvest index. And so this is this partitioning efficiency. And so now our best crops partition about 60% of all the above ground dry matter into the grain. And so the harvest index of wheat and corn and rice is about 60% under the best conditions. We think the theoretical upper limit's about 65%. So again, a little bit of improvement is possible, but certainly not a doubling. What wasn't improved at all during the Green Revolution was the photosynthetic conversion efficiency. And the reason probably is that all C3 plants do photosynthesis pretty much the same way. All C4 plants do photosynthesis pretty much the same way. And so there wasn't any genetic variation, natural genetic variation for breeders to select on. And so when they selected for yield, they weren't selecting for improved photosynthesis. But the capacity is there. And so as you've probably heard in farmers' fields under high input conditions, about 1% of the total sunlight is converted into biomass. We know the theoretical upper limit is about 5%. And so there certainly is the room for doubling if we can figure out how to improve the efficiency of photosynthesis. And so this is an equation. Are there empirical evidence that suggests if we increase photosynthetic efficiency, it will indeed translate to increased yield? And there's actually quite a bit of evidence for that, but I think the most intuitive is growing plants at elevated CO2. And so at the University of Illinois, we have what's called a FACE site, and FACE is an acronym for free air carbon enrichment. And so this is an experiment that we've been doing for the last several years, and this is the plant cassava. And so this is probably the only outdoor planting of a cassava in North America, but we can grow it for 4 years. And so what we do in a FACE study is these plants grow the entire season at 620 parts per million CO2. Out here in the soybean field, it's an ambient CO2 of about 420 parts per million. And so that increase in CO2 concentration increases the efficiency of photosynthesis by suppressing the oxygenation reaction of the primary carboxylase rubisco. And I'll talk a little bit more about that in the next slide. But the result of this increase in photosynthesis due to increased CO2 result in a 25% increase in cassava yield, a 16% increase in soybeans over the 14 years that we've done it, and then work that was done at other FACE sites around the world, 15% for rice and 20% for wheat. And these were done at lower CO2 enrichments than the experiments that we're doing in Illinois. And so the reason that CO2 increases photosynthetic efficiency is this enzyme rubisco. It's really a really bad enzyme. It's the most prevalent protein on Earth. It's the most prevalent enzyme on Earth by orders of magnitude. And what it's supposed to do, what we'd like it to do, is take this CO2 molecule, hook it onto a 5-carbon sugar, and then make 2 molecules of the 3-carbon sugar phosphoglycerate. And so this is where C3 photosynthesis gets its name, because its primary initial product is this 3-carbon sugar. But Rubisco evolved about 3.5 billion years ago. And it evolved in a time when there was no oxygen in the atmosphere. And so it evolved a mechanism, as it turned out, that can't distinguish that well between CO2 and oxygen, and they're actually competitive substrates. And so under current atmospheric conditions, about every 4th catalytic turnover of this enzyme is with an oxygen molecule. And this is really anti-photosynthesis. And so what happens is you make 1 molecule of phosphoglycerate, the 3-carbon, and then 1 molecule of this glycolate. Glycolate is 2 carbons. And so glycolate has 2 problems associated with it. One is, because it's 2 carbon, you can't put it back into the C3 cycle. And then secondly, it's inhibitory of 2 different enzymes in the photosynthetic reduction cycle. And so when plants encountered this, they had to look around metabolically and say, what can we recruit to deal with this? And what they recruited was a very complex, very energy-intensive process where glycolate is transported out of the chloroplast into the glycosome. Then 2 molecules of glycolate form a glycine. It's deaminated, CO2 is released, and finally 5 carbons went out, 3 carbons come back in as phosphoglycerate. For our purposes today, what we need to understand is this is really, really energetically expensive. So for every one of these oxygenation reactions that happens on every 4th turnover of this carboxylase, we're consuming 3 ATPs and 2 NADPHs. That probably doesn't mean very much to you, but let's look at what that means from the standpoint of the total energy currency of the plant. And so we put this information into a multi-layer canopy model. And so this is a model in which we can simulate in silico photosynthesis of an entire, in this case, soybean canopy. We parameterized it with data that we've taken out of a soybean field, and then we simply asked this multi-layer model, how much of the total ATP and how much of the total NADP is being used just to recycle this glycolate? And it's huge. In the middle of the day, it's 40% of the ATP and 30% of the NADP. And so this is not the side reaction. This is central metabolism, and it's really draining the energy resources of all C3 plants that are out there. We then took and embedded this canopy model into a agri-ecosystem model and asked the question, what is the potential yield drag that's associated with this? And so again, we've modeled this for soybean, and this is the US Corn Belt, and the US Corn Belt is responsible for growing about 40% of the world's soybeans. And what this shows, if we, in our model, if we compare the hypothetical case of no energy expenses due to photorespiration, so the oxygenation never occurred, versus what the real costs are under the current atmospheric conditions, you can see the yield drag is 35 to 40%. And so even small improvements in photorespiration, improving photosynthetic efficiency, this model suggests that there would be big returns associated with crop production. And so rather than waiting around for CO2 to get up to 660 parts per million or 700 parts per million, we thought what we would try to do is to install into the chloroplast a pathway that isn't as energy expensive. And so we tried 3. They all work to some extent. I'll talk mostly about one. And so one of the pathways is the glycolate oxidase pathway that exists in E. coli. And so we took that entire pathway. It's associated, it has 5 genes associated. We rearranged the DNA to match the DNA coding preferences of higher plants and put that into the chloroplast. And so you can see that it still decarboxylates and it still releases a CO2, but now it's releasing a CO2 in the chloroplast where Rubisco is versus in the mitochondria. Raises the CO2 concentration, and therefore, since they're competitive substrates, lowers the number of these oxygenation reactions that, that drive photorespiration. 2 other pathways were completely synthetic pathways, and so one of them we took glycolate oxidase, and so glycolate is usually converted to glyoxylate in the peroxisome, and so we moved that enzyme into the chloroplast. It uses oxygen as an electron acceptor, and so it makes a lot of hydrogen peroxide, which you guys know as one of the forms of bleach. And so in order to deal with that, because it's deleterious to proteins and other processes in the chloroplast, we had to stuff a lot of catalase in there. And catalase converts hydrogen peroxide into water and oxygen and makes it harmless. And then we put malate synthase in to convert gloxylate to malate, and then the chloroplast has enzymes to decarboxylate it in the chloroplast. The other pathway that we tried is very similar, except rather than using the plant enzyme, we took glycolate dehydrogenase from Chlamydomonas, which is a green algae, and put it in there. And the potential advantage there, it uses an electron acceptor of quinone rather than oxygen, and so it doesn't make this hydrogen peroxide, and so we don't have to put all this catalase in here. And so what I'd like to do this afternoon is just follow this last pathway through to field studies that we've done. The other thing that we wanted to make sure is that now that we have this energetically favorable pathway installed, we want to run as much of the glycolate flux through it as possible. And so the way we reasoned to do that was to genetically down-regulate the formation of the transporter that transports glycolate out of the chloroplast into the native pathway. And so essentially deactivating the pathway by preventing glycolate being introduced into it. And so this is a little bit technical, but it's one of the things that you have to show. You have to show that those genes that you put in are expressed, that they make the protein that you want them to make it, and importantly in our case, they target it to the compartment where we want it to go, which is the chloroplast. And so this over here just shows the transcript level, the RNA that's made from the transgenes that we put in. And so both the glycolate dehydrogenase from Chlamydomonas and the malate synthase that we put in from pumpkin, those weren't native genes, and so therefore these expression levels don't mean a lot, and so they're expressed hundreds and hundreds of times over something that's not there, and so, but nevertheless, they are expressed. And then to down-regulate the exporter, we used interference RNAi, or interference RNA. And so that just interferes with the overall translation of that message and prevents that protein from being translated. And so what that shows is that each one of these numbers are a different transformation event, and you can see in these three events when we included the RNAi that this transcript level goes very low. And then we do immunoblots to show that those transcripts are translated into protein, and so this is the leaf. We expect them to go in the chloroplast, and then this is the membrane fraction. And so what this shows is that, that the dehydrogenase is expressed in the chloroplast. It's expressed mainly with the membrane fraction. Malate synthase is expressed in the chloroplast, totally in the stroma. And then you can see with the RNAi that this glycolate exporter is down to very low levels. And so it worked. I mean, from the standpoint of expression, things were expressed and they went where we intended them to go. And so I posited to you that this is a more efficient pathway, and one of the ways to look at the efficiency of photosynthesis is to measure what's called the quantum yield. And so this is the number of photons of light that it takes to reduce the CO2 molecule to the level of a carbohydrate. And so theoretically, that is, in a C3 plant, is about 9. But photorespiration raises that up because every time it's doing an oxygenation, it has this big cost. And so what you can see here, here's the wild type, and here are various transformants with and without PLGG1, and you can see that there's about a 20% increase in the efficiency of photosynthesis measured as the quantum yield. And then our final tests, I mean, we did a lot of greenhouse tests, we did a lot of growth chamber tests, but what we really wanted to see, does this show through under agricultural conditions in the field? And so all of these experiments were done with tobacco, and the reason to use tobacco is manyfold. One is it has a well-defined genome, it's easy to transform, and it produces a huge amount of seed. And so in the first generation, we have enough seed to do a field test. And so this is a field test that we did in 2017. We repeated it in 2018 and 2019. And I just want to direct your attention to two things. One is this increase in quantum yield that we saw in the greenhouse is sustained in the field, and so you can see the wild type here, and you can see the with and without the RNAi for the exporter here. And again, we're seeing this 20 or 25% increase in efficiency. And then if we go down here and we look at the total above ground dry biomass, you can see, let's just focus on the total here. You can see that with the RNAi module in there, we're seeing 25 to 30% increases in the total biomass of the field. And so from the standpoint of growing bigger plants on the same amount of land, we've succeeded. Of course, we weren't out to make better tobacco. Having said that, after we published this paper in Science in January of last year, the first call I got was from a tobacco company, followed by 2 calls from cannabis companies. And so, you know, what we're doing now is we're trying to move this into seed crops, and we already have it in potato. We haven't done a field study, and we have it also in soybean, and we're trying to put it into some other plants. And so a year from now, we should know if this translates into yield. Another thing that I wanted to point out was one of the things that we really expected is that this alternate pathway should have its biggest benefit at higher temperatures. And the reason to think that, I showed this slide before and I talked about the soybean grown in the Corn Belt, but this agro-ecosystem model suggests that as you go further south into soybean growing regions, the losses approach 50%. And the reason is, in the model, it's parameterized as being warmer. And the reason that that is significant is this enzyme's ability to discriminate between CO2 and oxygen gets worse as temperature goes up. And so this just compares leaf photosynthesis as a function of temperature. And so what you can see is these are the wild type, these are 3 different transgenic events, and you see that they begin to separate where photosynthesis is doing better in the plants with this bypass reaction embedded at about 25 degrees. And by 40 degrees, they become statistically significant. I talked a little bit about our FACE project. The other thing that we have at the FACE project is the ability to warm canopies because we want to look at the interaction of CO2 and elevated temperature. But what we did here was just to use those heating arrays to raise the temperature. And so what you can see here is a tobacco plot that is underheating versus the control. And again, we have wild type, and then we have 3 independent transgenic events for this pathway. And so what we do is we measure the ambient temperature in this plot, and then we continuously add 5 degrees to it. And so, you know, throughout the day and throughout the season, whatever the temperature is, we're adding 5 degrees to it day and night. And we do that in quadruplicate. These are spaced around this 30-hectare site because they need to be next to transformers for the power pole. And so these experiments use a huge amount of power in order to warm these canopies 5 degrees. And so, again, if we return to this measure of photosynthetic efficiency by quantum yield, what you see again is, in the case of these events, and so here we have the ambient temperature and here we have the heated temperature versus the response to the wild type. And you can see by these different letters, they indicate whether the change is statistically significant. And so as the temperature goes up, you know, we are seeing the same statistically significant change that we see compared to wild type and AP1. And then when we add the 5 degrees, you can see again that all of these are doing better than this. We're able to do this twice in the season, and the reason to do this twice in the season is that we used a June planting date and a July planting date, and so the later planting date just has a higher background temperature, and we're looking at higher temperatures. And we can get 2 sets of experiments in one season. And so what we found in terms of biomass is this. In wild type, the 5-degree heating alone resulted in a 30% loss of above ground dry matter. And it was a bit bigger for the July planting, because of the warmer temperatures. And what you can see is each one of these independent transgenic events protects against that loss. And so, you know, I will admit that we were surprised by this big decrease in the wild type because tobacco is supposed to like warm temperatures, and we heated both day and night, and we think some of this has to do with the night heating. But what you can see is this is very significantly protective. And so I've given you one proof of concept example that improving photosynthetic efficiency can improve above ground biomass, and we have a reasonable expectation that that's going to translate into yield, and that's something that we're testing now. Here we assembled a table some years ago of 10 different redesigns that our in silico computer models suggest that if we make these changes, it'll show through in a significant way in the canopy. This work that I've talked about has been supported by the Bill and Melinda Gates Foundation, and the University of Illinois is the lead in this, but we have 3 international partners, 2 in the UK and 1 in Australia. And across those 3 sites, we're looking at 9 out of these dozen, and right now we have proof of concept of 3 of those, and we've made progress on all but 1 of them. And so this does seem to be a very promising strategy going forward. And so thoughts that I want to leave you with is that agriculture faces a major challenge, and maybe we have to as much as double productivity on a land area basis. Improving crop yield potential has to play a key role in this. I don't think anyone argues about that, and only improving photosynthetic efficiency is left in order to be able to do that. And then finally, there's a range of opportunities to engineer more photosynthetically efficient photosynthesis, and many of these potentially can be combined. And so we do think that this is a promising path forward. And let me just end by saying that the work that I talked about today was done by these very talented postdocs in my research group. So thank you very much.
Thank you, Professor Art, for illuminating the fantastic work you are doing on photosynthesis and how intriguing that is also, and also what kind of new areas that opens up for the future. We go now to a little bit lighter element, although lighter. It's a different element of the program. We've heard a lot on transitions and the need for that, and that also comes back in the music we are going to listen to. Let me introduce to you the Jasper Somsen Trio, they will come from somewhere, I guess, on stage. So they're standing over there, which consists of Jasper Somsen on contrabass, Rembrandt Friedrichs on piano, and Una van Geel on the violin, the alt violin. Yes.
Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in a forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level. We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in a forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in the forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in the forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore it's directly relevant for everybody.
We use parasitic wasps to study at the molecular level how they become males or females. In the animal kingdom, including humans, the differences between males and females are very pronounced and very diverse between animals. I want to know how that arises and how that evolves. So what fascinates me a lot is to understand how these big phenotypic changes that we see within the animal kingdom, how these are translated on a molecular level.
I'm Mehdi Habibi, and I'm interested in understanding the physics of everyday life phenomena. Crumpling a piece of paper is an everyday life phenomenon. What fascinates me about that is the simplicity of the phenomenon and the complexity of the underlying mechanism. By combining the knowledge of crumpled system with the origami structures, we are able to design robust material for real-world application. For example, for making a stent for medical application.
In the carbon cycle, one of the most uncertain aspects is the role of the forests, and we want to understand better how they take up CO2 and how this changes over time, especially in changing climate conditions. And by measuring oxygen and CO2 together, I hope to provide extra information to understand this forest sink better. So here you see measurements of CO2 and oxygen in the forest. I think it's a very relevant topic because it will directly impact how climate is changing, and therefore
for us at least a different connotation in flowing the different music styles sweetly into each other. Thanks a lot for that. I just, before this meeting, I ran into, let's say, an anonymous acquaintance, and he said, basically, the main reason why I come to your Dies Natalis is always the 3 young scientists which are appearing on stage. So we have again 3 amazing young scientists. May I ask to the floor, Mehdi Habibi, Evelien Verhulst, and of course also Ingrid Luijks. They come from 3 different directions, 3 different fields of science, and they will illuminate for you their fascination of fundamental science. I leave it up for you, the stage.
Good afternoon. So before we heard the very interesting talk by Professor Oort on photosynthesis research, and like his, also my research focuses on photosynthesis on a scale of forests like this one we see here in Finland. And I look at this from the atmospheric perspective. And what fascinates me is that this process that occurs in a single leaf of a plant, or a tree in this case, we can observe it back in the atmosphere. And this is also important because these forests and the global biosphere in general takes up CO2. And this has an impact on climate. So for climate, it is very important how much CO2 remains in the atmosphere because that drives climate change. And these forests provide a great service by taking up a lot of this CO2 actually. And CO2 concentration in the atmosphere is not constant, but it varies a lot, as we can see here in this computer simulation. So here you see emissions taking place in the industrialized regions in the world. For example, Europe, but also in the US and China. And all those emissions are transported by the atmospheric circulation and weather systems that you see here in the simulation. So they are transported initially mainly in the latitudinal horizontal direction. But after the course of a full year, the complete atmosphere is completely mixed. And you will see that the CO2 concentrations will even increase in the South Pole, completely isolated from the direct sources. And then photosynthesis starts to kick in when spring starts. Like we observe currently outside, the leaves are forming back on the trees, and by this process, they take up a lot of CO2. So you can see it obviously here that the concentrations are drawn downwards in the spring and summertime. And this is very important because with this process, less CO2 remains in the atmosphere. And this is everything all based on computer simulations, but we also observe this from a global network of stations. So these are all the locations in which CO2 concentrations are observed in the atmosphere. And these measurements are made by international colleagues at universities and research institutes. And one of the most famous stations of all is the Mauna Loa station, which was where the first measurements of CO2 were taking place from already 1958 by Ralph Keeling. And you can see this graph here. This is focusing only on the last 3 decades. And you can clearly see that the CO2 concentration is increasing with time, as we all know. But at the same time, you see very clearly the seasonal pattern, with lower values in the spring and summertime and higher values in the winter. And what my research then focuses on is how much actually is taken up by the biosphere. And we can observe the same signal actually in oxygen. So if we measure oxygen, initially you would only see this. So the oxygen concentration is very constant in time at 20.95%. But if we zoom in further, we will actually see the opposite signal as we see for CO2. So the oxygen concentration is actually decreasing with time, together with CO2, and this is because the process of fossil fuel combustion, of course, uses oxygen in this process, and therefore the concentration is going down. At the same time, you also see the seasonal pattern from the biosphere. So when the trees take up CO2, they produce oxygen. Now, and as you saw before, the oxygen concentration is still very constant at 20.95%. So this drawdown of oxygen is not something to worry about, because it's really only if we zoom in to the very final digits that we find this number. And to be able to make these measurements, we need to make very precise measurements, because the variations that you see here are in the order of 1 part per million. So it's one part per million over a background concentration for oxygen, which is over 200,000 parts per million. So you can imagine that to observe these very small changes, you need to make very precise measurements. And the variations for CO2 are in the same order of magnitude, but their background is only 400 parts per million. So therefore you can say that making atmospheric oxygen measurements is about 500 times harder to do than for CO2. And now this signal we see in Mauna Loa, which is a mountain on an island in the middle of the Pacific Ocean. And even there you see the signals of photosynthesis. But in order to learn more about it, of course, you need to go where photosynthesis actually happens. So that's what I did in my research. In 2018 and 19, I went to Finland to measure CO2 and oxygen in this site called Hytiala in the middle of the boreal forest. And this is a very important region, actually, in climate change, because temperatures globally are increasing, as we all know, but especially those boreal regions are very sensitive to this, and also temperatures are increasing much more rapidly there than anywhere else. And this site, Hytiala, is also very convenient because it has already many facilities. So you can see here a 125-meter tall tower from which we can actually draw air down and we can measure it downstairs in our laboratory. So what do we want to learn in this study? So as Professor Oort already mentioned, the forests are a very important sink for CO2. So globally, they take up about 25% of all the emissions that take place from fossil fuel combustion. But this net sink consists actually of 2 much larger processes. So that's photosynthesis on the one hand and respiration on the other hand. And if we then look at the concurrent signal of oxygen, it's in the exactly opposite direction. So those 2 large fluxes of photosynthesis and respiration will in the end determine how much CO2 will remain in the atmosphere. So photosynthesis is expected to increase with increasing CO2 levels.
But on the other hand, also respiration is expected to increase due to higher temperatures. And the balance between these two large fluxes is quite unknown and will be very variable, but it will be important to know how much the climate will actually change in the future. So that is why we need to find more information, because if we only observe the CO2 concentrations, we will never be able to distinguish these two processes. And in my research for that, I use oxygen, because it's linked directly in the process level. So if you look to the basic equation for photosynthesis, which is shown here, when pure sugars are formed by combining CO2 and water, oxygen is produced at the same time. And if you look then to the exchange ratio of oxygen over CO2, you will find a ratio of exactly 1.0. But of course, that's not all there is to it, because there's not only pure sugars that are being formed. There's also other elements involved in the reactions, like minerals, and also other components are formed. So this value of 1.0 can also show very slight variations, similar to what we saw before for the oxygen concentration. So by measuring CO2 and oxygen together, we hope to find these very small variations to inform us on how these two large fluxes will change in the future. So that's what we tried to do in Finland. And now it would have been very easy if I would just take my oxygen analyzer to the forest and put it there and measure the oxygen concentration. But unfortunately, it's not so easy to measure oxygen. So this instrument that you see here, you can buy it commercially. It consists of two small fuel cells, which will produce a current based on the amount of oxygen that is in the air. But actually, to measure these very small variations that I showed, you need to stabilize everything around it. So in practice, it was not just taking the thing, but it looked a bit more like this. So we had much more materials which we need to transport into the forest. And finally, the oxygen analyzer is only a very small part, which you see just behind my hands there on the screen, of a much larger instrument that we developed during my PhD at the University of Groningen. And it consists of much more elements. So it has pressure stabilization, temperature stabilization, flow stabilization, and everything controlled and in comparison to standard gases that we take along with us to compare the values to. And then in the end, we can actually measure. And then we see what a day and a night looks like in Hyttala in Finland. So you can see here that during the nighttime, the CO2 concentration, which is the bottom panel, the CO2 goes up and oxygen goes down. Whereas during the daytime, when photosynthesis is active, oxygen is produced, oxygen goes up, and CO2 is taken up and it goes down. And these signals are all mixed inside the atmosphere, and actually they are mixed in the lowest part of the atmosphere, which we call the atmospheric boundary layer. And this layer is much shallower during the nighttime, which also leads to much larger signals. Whereas in the early morning, when the sun rises, this layer starts to grow, so the signals are mixed in a much larger volume. But also, when the sun starts to shine, photosynthesis kicks in. So there's many processes that occur at the same time, and by combining these measurements together with other measurements, for example, on the vertical structure of the atmosphere, using weather balloons, we can actually distinguish all the processes. So to wrap up, oxygen can provide us with much more information on the drivers of the CO2 signal, but it's not so easy actually to measure it. And then the atmosphere integrates all the different signals, and by combining different types of measurements together with atmospheric models, we hope to learn something about how the forests are taking up CO2 and how this varies, and how they will continue to take up CO2 in the future. Thanks a lot for your attention.
Look around you. Can you see the differences between your own sex and the other sex? Of course you can. We all look different. Facial hair, muscle growth, body shape, these are all biological characteristics of males and females. Human males and females. But also in the animal kingdom, there's a wide variety of differences between males and females. Take this classical example of the Mandarin duck. The female has a very dull brownish appearance, but the male is brightly colored. And also within insects, we see the same thing. If you would look at these butterflies, do you think they belong to the same species? Well, actually they do. It's the female, a bit dull greenish in color, and a bright orange male. Well, we know already how these differences evolved, and it has to do with sexual selection. So if we take again this example of these butterflies, and we have a population in which males have a dull orange color, and you still have the same dull greenish color in females, but there's one male that has a bright orange color, then of course he would get noticed. And females would think, whoa, he stands out so much, that is my partner, I want to mate with him. So of course that's then what happens, they mate, and if they mate, what do you get? Of course you get offspring. So if we have that female and that male and they produce offspring, what you would see is that actually the females would have the preference for bright colored orange males, just as their mothers did, and the males would take the traits from the father and have the bright color orange. So what you would see is that actually between males and females these traits would get more and more exaggerated. And we see this happening within nature a lot, and it actually happens on a very rapid scale, so it evolves quite rapidly. My fascination is actually with how does this evolve so rapidly? How can it take place? And especially, I'm interested in looking at the smallest level, at the molecular level, just as Ingrid before me was interested in the smallest details in changes in CO2 and oxygen. So I want to understand how can these rapid changes in male and female appearances, how does this happen on a molecular scale? Well, for this, I use a wasp, a very tiny wasp. But the funny thing is, this wasp has a number of characteristics that makes it very useful for my research, because it has differences between male and female characteristics, like for example the sex pheromones. Males and females of course produce different sex pheromones, but also perceive sex pheromones in a different way. The pigmentation of the legs and the antennae is different between males and females, with females having a dark colored hind leg and dark colored antennae, and in males this is bright yellow. And then the wing size. Females have big wings and can fly, but males have very short wings and actually cannot fly. So we know in insects which gene is actually at the bottom, or is underlying all these differences. And that gene is called double sex. And double sex is a very specific gene in that it can actually be encoded into a female-specific form and into a male-specific form. And double sex, when it's encoded as a protein, we call it a transcription factor. And transcription factors can switch on or switch off other genes. And in this case, if we take the male form of double sex, it can switch on the genes necessary for male development and switch off the genes necessary for female development. So if we take, in this example, the gene for shorter wings, then in front of it would be a site in the genome where double sex as a protein can bind to. And when it binds there, in this case, because it's the male double sex protein, it will actually activate the gene for shorter wings. And then you get shorter wings. Well, this is all, of course, on a very basic level. And for many of these things, we actually don't know what are the genes that encode all these differences we see between males and females. So we don't know them. We don't know the genes. So one way, me and my team study this, is to actually take double sex out of the equation. And if we take double sex out of the equation, we can see what happens in the development of males and females, and then understand which genes are underlying these. We can do that by injecting a specific compound into adult wasps, in this case, a male. And imagine that. They were so tiny, we can inject them. How cool is that? So we do that in the same way, actually, as Professor Oort has already been saying, we use RNA interference. And we can do this in different developmental stages, ranging from tiny eggs to early larvae, late larvae, pupa, and adults. And depending on when in the developmental process we actually silence the gene, we can see differences. So if we again go back and we inject the compound to silence double sex, and we take the example again of the gene for shorter wings, what you would expect is, of course, that if double sex cannot switch on the gene for shorter wings, is that the wings would grow, right? So then you would assume that those males, after we've silenced double sex, would actually have bigger wings that more resemble those of females. And that's exactly what we see. So if we take the normal female with the bluish outline, and we have the male on the other hand with the red outline, then if we silence double sex in the male larvae, indeed, in yellow you can see, we have a wing size that is more or less intermediate between that of the male and that of the female. And by doing this in different stages during development, so injecting it very early, so silencing double sex very early in development, compared to doing it later in development, we actually see we can have different effects. So the earlier we silence double sex in development, the stronger the effect is and the longer the wings get. So I've shown you now what happens in the wings, and of course, by studying the phenotypic traits, we still don't know the genes. But we have another technique for that. So if we take all the genes that are being expressed in the males, in the females, and we compare those to the intersex that we've created, we can actually see which genes are being expressed differently between males and females in the intersex, and then couple the genes with the phenotypes we see, and then get an idea of which genes double sex is actually regulating. So now I've shown you how we do that for wing size, but we're also interested in how males and females perceive sex pheromones. And of course, that takes place in the brain, in a specific brain area called the antennal lobe. And the antennal lobe is the brain area where odors are processed. And we know from research that the differences in the female brain region compared to the male brain region is quite pronounced, in the sense that females have more odor processing particles in the brain than the males do. And if you then take a closer look, in this case you see the example where we've looked at the female brain region and compared it to the male brain region. And we see in females the blue outline, that's a female-specific region, and that's absent in the males. But if we silence double sex in the late larval stage, we actually get males with a female brain region. How cool is that? Super nice. So all in all, by combining the information on a phenotypic level with the data that we get back from looking at the gene expression, we get an underlying sense of what double sex is regulating on a molecular level and get a better understanding of how these differences evolve. So I want to highlight that when 20 years ago researchers started studying sex determination, it was just for their curiosity. I'm also studying this because of my curiosity. I want to understand how it works, especially at the molecular level. But now, with of course, the mosquito populations getting out of control, biocontrol that we need to apply, in many cases it's only one of the two sexes that we want to target. For example, if we want to target mosquito populations, we only want to target the females, not the males. So knowledge of how double sex is regulating sex-specific genes, or in this case, female-specific genes, we can actually modify them and only target the females and not the males. Nobody would have thought of this when they started it 20 years ago. So for that, for my take-home message for you, I want to stress again, and related to what Arthur Mol already said, fundamental science is essential to satisfy our curiosity, mine as well, but also to understand the world around us and to protect it in that way, but also to be able to solve problems before they even arise. Thank you.
Good afternoon. Almost all of us are familiar with crumpling a piece of paper. If we look at the nature, we can also find crumpled structures at different length scales, from DNA packed in a viral capsid, like coronavirus, to crumpled wings of a butterfly coming out of a cocoon. In the next few minutes, I want to introduce you to the world of crumpled metamaterials. If we look at the history of human, we see that humans have used different material, and this sort of material has revolutionized our life in a way that different era of civilization are named by these material, like stone, bronze, and silicon. Some scientists believe that the next revolutionary era in the history of human could be the era of metamaterials. Metamaterials are materials with unusual properties, and they have these sort of properties because of their structure rather than their composition. From everyday life, we know when we stretch a piece of rubber, for example, as you can see in this movie, it starts to shrink in the perpendicular direction. And the amount of shrinkage can be quantified by a parameter which is called Poisson's ratio. The Poisson's ratio for rubber and other sort of natural material is always positive. But we can design structures like this toy that they can expand by stretching. There is another example here in this movie. It also shows expansion by stretching. If we calculate the Poisson's ratio for this sort of materials, we find that the Poisson's ratio is negative. And this is an unusual property in natural material. In my research team, we are making different meta structures, and we are trying to link the curvature of these structures to their Poisson's ratio. As you can see here, I have two examples, a dome-shaped structure and a saddle-shaped structure. And by linking the Poisson's ratio to the curvature and understanding this relation, we are able to program the shape of these objects. By programming the shape, then we are able to design structures like the structure that can be adapted or matched to a specific curvature, like curvature of an apple, for possible applications in soft robotic. Or the other example is a mirror, which was designed by one of my students, and it shows adjustable focal point. All these structures can be made by 3D printing. But there is another way of designing mechanical metamaterial, and that's origami. So origami is not only the art of making or folding paper. This is a science for designing new material with new functionalities. As you can see in this movie, by stretching the origami, it can expand. So it shows negative Poisson ratio. These sort of structures have shown lots of application in different areas of science and technology. For example, for making foldable solar panel for aerospace application, or foldable stand for medical applications. But there is a problem. Since they are highly ordered, they are very sensitive to dislocation. As you can see in this example, by introducing a dislocation in the structure, their mechanical functionality can be strongly hindered. So the main question here is that how we can design a structure with functionality and reduce the sensitivity to randomness and dislocation for real-world applications. My answer to this question is based on using crumpled structures, because crumpled structures are made of disordered fold and creases, so they are inherently insensitive to randomness and dislocation. They have other interesting property that we can use for designing new materials. For example, as you can see in this movie, by stretching a crumpled piece of paper, it also expands in the other direction, which shows a signature of a negative Poisson ratio. The other interesting property is that they can be adapted to any curvature. That's why we use aluminum foil to wrap different objects. The other interesting property of crumpled system is that they can show high stiffness. If I crumple a piece of paper by hand and measure the amount of force I need for compaction, you can see in this plot that the forces start to increase by reduction of the size, and at some point it diverges. This means that this material can be very stiff. And if we look at the structure, it is still composed of 70% void in the structure, which means that they are super light and very stiff. So they are excellent candidates for making lightweight, robust, and stiff materials. But you may ask how we can design this sort of completely disordered structure to get a specific functionality. To answer this, we need to look at the anatomy of a crumpled structure. Here I have two examples. One of them is made of crumpled aluminum foil. The other one is a cross-section of a crumpled rubber sheet. As you can see, there are differences in the morphologies. Specifically, the aluminum foil has denser outer layer respect to the inner layers. These changes in the structure and morphology can contribute a lot in the mechanical response. So by changing the morphology, we are able to tune the mechanical property. And there are simple parameters like plasticity of the material, friction, and adhesion between the layers that can help us to tune the morphology and consequently tune the mechanical response. So now we have found a way to design a mechanical crumpled system with desired mechanical property. But then the question would be, is it possible to use this sort of knowledge to make more robust origami-like structures? Because we know that the origami have lots of application with lots of functionality. In order to do that, we have combined the knowledge of origami science with the knowledge of crumpled system, and we have designed new structures. I have one example here. We call them origami-like crumpled structures. As you can see here, this structure shows similar properties like the origami that I have shown. So it also shows negative Poisson ratio effect. And it is made of a crumpled unit cell, which is repeated in a lattice of a specific symmetry. By this sort of design, we can make origami-like structure with similar functionality, but at the same time, we can reduce the sensitivity of the origami structures to noise for real-world applications. If you are still looking for more tangible applications, I have several possible applications for this sort of fundamental research. By understanding the physics of a crumpled paper, we can design new materials, like new sandwich panel for different applications, which are made of crumpled materials. The other possible application would be designing effective shock absorber. For example, designing a new crumpled zone for the car to effectively absorb the mechanical energy. Or we can even think about new sustainable packaging method to reduce the amount of plastic that we use every day for packaging purpose. As I mentioned at the beginning of my talk, nature is aware of the robustness and efficiency of crumpled system, and we can find interesting examples in the nature. By understanding the physics of a simple crumpled paper, we might be able to shed light on this sort of complex biological example as well. So with this, I would like to thank you for your attention.
Thanks a lot, Ingrid, Evelien, and Mehdi, for fantastic presentations and a very inspiring type of research. Some of the research you wouldn't expect here at Wageningen University, but we still do it. One big hand again for these three magnificent people. And I would like to call upon, we are almost there, ladies and gentlemen, so if you get thirsty, 5 more minutes. To Professor Erik van der Linden, who will hand out the research award, which we always do to one of our young scientists for an excellent publication. And Erik was chairman of the jury for this year. Erik, floor is yours.
Thank you, Mr. Rector, dear ladies and gentlemen. University Fund Wageningen wants to support and highlight work of gifted young researchers and therefore awards a prize yearly to a researcher who has published an original and excellent scientific article in the calendar year preceding this ceremony. The entire scientific community of Wageningen was invited to submit nominations, and a jury composed of Professors Bas Swaan, Alfons Oude-Lansink, Drs. Daan Swartz and Suma Munte, one of the winners of last year, and myself evaluated all nominations. And we were very impressed by the quality and creativity in all of the nominated papers, and we thank all the nominators for submissions. The winner of 2020 has been chosen, and I feel very honored to be giving the Research Award of the University Fund Wageningen to this excellent young researcher. Notably, this person will receive €2,500, a statue of the Wageningen tree, and a certificate. And without further ado, I can announce that the winner of the University Fund Wageningen Research Award 2020 is Dr. George Navarro Munoz, for the article entitled Computational Framework to Explore Large-Scale Biosynthetic Diversity. Thank you. The article appeared as a cover article in Nature Chemical Biology. It presents a new bioinformatics tool, allowing rapid analysis of large data sets. The data sets in the article are about production routes of secondary metabolites, important for understanding how microorganisms and plants interact with their environment and, for example, also for developing anti-cancer drugs. The jury applauded the fact, and applauds, the fact that the article is very readable for scientists that are laymen in the field, and that the authors also used the tool to predict new production routes and experimentally verified these as well. Furthermore, the jury is very much impressed by the fact that the newly developed methodology has already been taken up and implemented in other high-impact articles written by others. And the jury therefore also expects that the work will form a cornerstone for data-driven approaches in the field. Now, unfortunately, the winner couldn't be present because he is in Mexico, but luckily, I've understood his nominator, Dr. Marnix Medema, is present to receive the award. And I see somebody coming up, so I guess that must be Marnix. Right? If you want to join me on stage, that would be great. And then I think I can... two awards.
Okay. Yes, all honor to Jorge. He was so incredibly surprised when I emailed him. He was indeed on holiday in Mexico, very well-deserved holiday after so much hard work. And he was completely flabbergasted with hearing this news, but it's incredibly well-deserved. He came to Wageningen a couple of years ago with a PhD in electrical engineering. And within a few years, he really became an expert in bioinformatics, in microbiology, in biochemistry, in chemical biology. It was really very impressive how quickly he grasped all the details of all these various fields and was able to spearhead such an interdisciplinary efforts with scientists around the world in various disciplines. So very well deserved, and congratulations to Jorge.
Thank you for being here in his stead, and the jury also wanted to extend the best wishes to Dr. Navarro Munoz in his further scientific career. And with this, I'd like to give the word back to the rector. Thank you.
Thank you, Professor van der Linden, for acquitting yourself of your duty of handing out this very fine prize to a young scientist who did such a magnificent job in doing his research. With this, we have come to the end of our 102nd Dies Natalis. We are up for another year, which will be full of surprises. We are in the middle of a big surprise, of course, which we didn't expect to happen. But probably in 10 years' time did this big surprise, the coronavirus pandemic perhaps, will only see a small rippling in a major continuous growth, not so much quantitatively, but especially qualitatively, of our university and research organization. Thank you very much all for being here, and join us for a drink downstairs. And with this, I close this meeting. Thank you.
Keep running to your arms, keep longing for your kiss. Keep running to your arms, keep longing for your kiss. Keep wanting you, keep wanting.